Thank you for that nice introduction. The first rule of public presentation is to know your audience. And usually I know my audience. But here I'm not sure how many of you are scientists. So if you're not a scientist, can you raise your hand? I'm actually a scientist, but I'm just showing you how to do it. Okay. All right, that's helpful. So today I'm going to talk about innate immunity, which I consider to be cutting-edge science, which unfortunately cuts all ways. Of course, I would like to start by thanking the people who brought me here to Belo Horizonte, especially Mauricio and Ricardo, who have been fantastic hosts over the course of the last year. And I'm sure to see more of all of you in years to come, as Ricardo explained. I also want to start by thanking the people who work in my laboratory. These are my postdocs and students who have over the years helped us publish papers. This doesn't have my old students, the ones who have left, because there's not really enough space in the slide. I've gotten old, and there are a lot of them. Also want to thank Ricardo. His laboratory has been working not only here in Belo Horizonte, but he has a group at UMass, and Marco, of course, who was part of our original collaboration. Bernardo Franklin, who's no longer in Ricardo's lab, but is now in Michael Lotz's lab in Bonn, was a fantastic student to have. And then I have a lot of colleagues and collaborators from UMass and other places across the world. And as a scientist, I don't think people realize how important collaborators are, because we can't do it all by ourselves. So it's very useful and enriching and fulfilling to be able to work with such talented people. So we like to say that the innate immune system is a double-edged sword. That is to say, on one side, we have tissue damage that leads to pathological repair as a result of innate immune dysfunction. And on the other side, we have a rapid repair. So we have the defensive and the offensive side to innate immunity. And really, what we would strive for is to have the right balance, either normally, through our normal innate immune defenses, or using therapeutics. So we have the innate immune system and the ways of protecting people from damage. And of course, when there is imbalance, damage can occur. Here we see a patient with systemic lupus erythematosus and another patient with crippling rheumatoid arthritis. And these are examples of the innate immune system gone awry. I'm an infectious disease specialist, really, and as an infectious disease specialist, we're particularly concerned with the response to pathogenic challenge, either from viruses, bacteria, fungi, or parasites. The innate immune system, as I'll explain in some more detail in a few minutes, is the early response to the challenge with these pathogens. Later on, we have what we call adaptive immunity, or the adaptive immune response. Those are the antibodies and the T cells that come to fight pathogens. But ultimately, both the innate immune system and the adaptive immune system have the same goal: to eliminate danger, those things that challenge our body and cause disease. Innate immunity I like to think of as the immunity that we are born with. This is how we arrive in the world. It's a very important part of the immune system. When we consider why do we need innate immunity is the immunity that requires no pre-exposure to a pathogen. We need this because pathogens multiply very fast. So typically, bacteria will double every 30 minutes. So let's take an example where you're brushing your teeth in the morning and bacteria from your mouth get into the blood. This happens to all of you every single day, at least those of you who brush your teeth. This happens every single day. So bacteria would double every 30 minutes. So in just 3 days, you'd have 2 times 10 to the 43 bacteria in your blood. This is clearly not compatible with life. Adaptive immunity requires 3 to 14 days to respond. So we need the innate immune response for these types of challenges. In fact, innate immunity is a lot more interesting than adaptive immunity, as you can see from the response of our students in Massachusetts listening to a lecture on adaptive immunity. The innate immune system has an arsenal of defenses that are truly powerful in fighting disease. These include fever, inflammation, phagocytosis, and complement proteins. Phagocytosis by all sorts of important cells, like macrophages especially, and neutrophils. So let me just start and tell you a little bit about fever. I find this to be a particularly interesting topic. People debate about fever. What's it good for? And this debate is ongoing and unresolved. But I propose to you that fever is good because it keeps mammals from wasting precious energy during infection. And it can be obviously deleterious as well. For example, we've seen patients in our intensive care unit who have very high fevers and have convulsions. And that's not a good thing to have. Matthew Kluger in the 1960s did what we consider to be classic experiments with fever. And to do these experiments, he used animals that are known as quaquilotherms. These are animals that don't regulate their temperature like you and me. They do it by, when they want to warm up, they walk into the sun. When they want to cool down, they walk into the shade. So the animal that he used was the iguana, and he infected them with a bacterium known as Aeromonas hydrophila, a very powerful pathogen. And you can see how what he did for his experimental setup, he had a cage, and one side of the cage had rocks that were warm and lights to keep the animals warm, and the other side of the cage was in the dark and had no warm rocks. So what he did was he injected Aeromonas into these animals, and the infected animals would increase their body temperature by moving to warmer locations in their cages. The first experiment involved 12 animals. 11 of them survived, and the only animal that died was the animal that did not move over into the warmer territory. So when they moved over to the warmer side of the cage, their temperature went from about 30 degrees Celsius to 40 degrees Celsius. This is quite a big difference. And when he took these animals and he treated them with salicylate, or aspirin, this prevented fever in 7 of the 12 iguanas, and all 7 of these animals died. So it looks like in this case that fever is a very good thing. Now, inflammation is a word that comes from Latin, inflammare, and it means to set on fire. And it's part of the complex biological response the body has to pathogens, damaged cells, that is host cells, or irritants. So inflammation is a protective immune response. It's an attempt by the body to remove injurious stimuli and initiate the healing process. And on the net whole, we believe that inflammation is a positive response. It's a response to trauma that has evolved over millions of years to keep us alive and healthy. Celsus, the Roman physician, was the one who first defined the signs of inflammation, which we list here on this side, the 5 cardinal signs of inflammation: rubor, color, tumor, and dolor, redness, heat, swelling, and pain. And at the end here we have function lossa, or loss of function. And we can see Bill Clinton here has gone through all 5 of those stages. This is what inflammation looks like. I'm sure all of you know what inflammation looks like. Here is infected skin. You can see that it's red, swollen, it's painful, and it's hot. And of course, there's loss of function. Inflammation is mediated through white blood cells that course through your blood. We can do that one again. And you can see the white blood cells as they go through the venous system here. And if we take an infected area, we can actually watch white blood cells traffic from the bloodstream into an area of inflammation, and it's there that the white blood cells will mediate the inflammatory response. Therefore, it's not like this is a bad thing. So here's an example of a phagocyte that's swimming around in a pool of bacteria. These happen to be Staph aureus. And this is a neutrophil. And you can see it's following that bacteria there with the design of eating that bacteria. And once it engulfs that bacteria, it can then take it and put it into the proper intracellular compartment where it releases enzymes and reactive oxygen products and kills the pathogen. A very, very important immune response. So phagocytes internalize microbes and kill them, but in addition, they secrete soluble molecules, we call them factors, that recruit and activate other effector cells that in turn kill microbes. So here again is a phagocyte encountering E. coli and being activated to release all sorts of inflammatory mediators. Now, one of the things that I'm most interested in is known as cellular signaling. So in addition to phagocytic receptors, the innate immune system has an army of signaling receptors that alert the body to the presence of pathogens. Here we see a virus flooding in and one of our receptors shouting alert to the immune system. One of the most incredible things about the innate immune system is looking at its evolutionary development. The phylogeny of innate immune responses demonstrates very clearly that these are very, very highly conserved evolutionary proteins that are responsible for the innate immune system that began with sea sponges, sea anemones, insects, worms, mollusks, echinoderms, all around 450 million years ago. Really incredible to think that innate immune defenses could be preserved over such a long time, but demonstrating also the central importance of innate immunity to survival. Now, one of the very many stories in innate immunity that are worth discussing is one that began with Christian Nusslein-Volhard, shown here. Christian Nusslein-Volhard won the Nobel Prize in 1995 in medicine for her studies that she did on Drosophila development. Nusslein-Volhard had no interest, and still has no interest, because she's still very much alive, in immunity whatsoever. She's interested in the genes that are responsible for development. And Nusslein-Volhard did not study human development. She tried to simplify things by studying Drosophila, the common fruit fly, the same animal that likes to eat your bananas. And I've shown you what a normal Drosophila looks like here on this slide. It has a dorsal surface and a ventral surface, and these are known as denticle bands. And these are where the hairs will ultimately, and feet will ultimately sprout. Here's what the animal breathes out of, known as the operculum, and here's the gastrointestinal tract. So that's the animal. You can see it's very, very highly organized. And Nusslein-Volhard wanted to know what were the genetic instructions that told cells that they belonged, say, in the stomach or in the operculum, as opposed, or the dorsal surface or the ventral surface. So she was looking for actual mutants, and mutants are a big theme in innate immunity. She was looking for mutants in which the instructions had gone all wrong. So this is a fruit fly, and you can see it does not look like this beautiful fruit fly here. It's this ugly fruit fly. And that's because all the cells believe that they belong on the dorsal surface of the animal, or the back of the animal. And so it doesn't develop normally. And the story is that Nusslein-Volhard was looking underneath a microscope, and she found this mutant, and she said, Toll. So Toll is a German word, and one of my ex-postdocs, Eike Lotz, this is still unpublished work. He defined Toll in English. Amazing, bodacious, cool, quirky, crazy, frantic, furious, great, like blazes, mad, madcap, super wild, wicked, and wow. It means all of those things. In Portuguese, it means bacana, something like that. Or legal. So, you can tell she was really very, very excited. And Bruno Lemaitre, a student from another university in Strasbourg, read her paper. And he said, boy, Toll sounds interesting to me. And then somebody cloned Toll, and Bruno read that Toll actually had similarities to a human gene that was important in inflammation, known as the interleukin-1 receptor. So Bruno got these Toll mutants, and he challenged them with different microorganisms. And this animal has been challenged with a fungus known as Aspergillus fumigatus. And what's happened to this animal is that the Aspergillus that the animal was exposed to have killed the animal dead. It has no immune responses whatsoever. Now, flies only have an innate immune response. They don't make antibodies, they don't have T cells. And look at this, these hyphae of Aspergillus that have killed this animal dead. And Bruno concluded from this that this was an important innate immune response. And he went on to suggest, along with his mentor Jules Hoffman, that there were orthologs of toll in humans that might be similarly important. And indeed, we now know that humans have 10 of these orthologs. They're known as toll-like receptors, or TLRs, and there are also 4 adapter TLRs. Mice have 13 TLRs. But one of the most interesting animals is the purple sea urchin. Now, the purple sea urchin has 222 TLRs. These animals live 60 to 70 years in estuarial waters that are filled with bacteria, and they have no acquired immune system. So we always say, the fruit fly, it has innate immunity because it only lives 3 weeks, and therefore it doesn't need to have an acquired immune system because it would never live long enough to make an antibody. But this animal lives 60 years and lives off of its toll-like receptors. I only wish I could do the same thing. So TLRs, we now know, are really very important for their ability to recognize microbial products from all sorts of bacteria and viruses and fungi. And they activate the innate immune response. Now, those of us who work in the toll field are absolutely amazed how this recognition of toll has changed the world of immunity. In the last 10 years, thousands and thousands of articles have now been published on toll, and it's opened up a new area of innate immunity, and all sorts of pharmaceutical companies now work in drug discovery. But not everybody actually recognizes this. In the United States, we have a politician named Sarah Palin. Many of you might have heard of her. She ran for vice president with John McCain, and fortunately she lost. But here's what Sarah Palin has to say on this story.
Some of these pet projects, they really don't make a whole lot of sense. Some of these dollars, they go to projects
that's not going to
have
work.
nothing to do with the public.
I have another idea.
Things like fruit fly research in Paris, France. I kid you not. Can't believe that. Can't believe those stupid scientists.
Sort of reminds me of my mother. She always is asking me why I don't go back and become a doctor. Anyway, Sarah Palin doesn't appreciate fruit fly science because I can't remember why, but she doesn't. But toll-like receptors, which came from fruit fly research, are really key drivers of inflammation and innate immunity. So there are 10 of these TLRs, as I told you before. Many of them recognize microbial products, but also when tissues become damaged and inflamed, they release molecules that also engage toll-like receptors. And that's very important. If you have a heart attack or a motor vehicle accident, toll-like receptors will become activated, immune genes will be turned on, and immune and inflammatory effector mechanisms will occur so that you can become cured. Now, I'm going to tell you a little bit of work that I do. This is the type of work that we do in our lab, and also we do in Ricardo Gazzanelli does in his lab, and really worldwide, about how toll-like receptors are involved in inflammation. And a lot of this is mediated through molecules that we call cytokines. And there are 2 cytokines that have a central role: TNF, or tumor necrosis factor, and interleukin-1. These are very important for immunity and inflammation. The main source of these molecules is a cell known as the macrophage, the main source of TNF and IL-1. And when the macrophage is activated by certain microbes, it releases TNF and IL-1 to activate endothelium. This causes endothelium to become very sticky so that immune cells that are floating by in the blood will stop and pay attention to whatever is happening nearby. It also helps other cells that activate the acquired immune system to mature. It recruits neutrophils to eat bacteria and other microbes, as in the movie I showed you a little bit earlier, and lymphocytes to become activated to produce antibodies and other effector molecules, and themselves to go after infections and kill bacteria and parasites. So cytokines are very important, but they mediate both the beneficial and the pathological effects of innate immunity. Again, this balance between protection and damage. So when we think about that, we realize that the production of cytokines and the activities of cytokines must be highly regulated. If they were not highly regulated, we'd all be in trouble. So consider the types of diseases that biological scientists have to study today, or the course of microbial diseases, and what the challenges might be. For example, most infections that you and I get in our daily life are actually asymptomatic. Most of the things that we encounter in the world will never have a symptom. The pathogen will increase over time, the innate and acquired immune system will become activated, and the infection will resolve, and we'll never know about it. Of course, lots of times we get sick, and the immune system is turned on. We have disease, for example, influenza. We feel sick for a little while, then things get better, we're recovered, and we're cured. Sometimes things don't work out so well, and infection results in death. And this happens, for example, in malaria, a disease that Ricardo and I study a lot. About 1% of patients who become infected will ultimately die. If patients have no pre-existing immunity to malaria, up to 60% of patients will become infected if they're not given drugs. And then there are the infections that occur. They seem to resolve, then they get worse, then they get better, then they get worse. We think that there are a lot of, for example, tumors that are started from a viral source that are like this. We think of some of our inflammatory diseases like arthritis that get worse and better, that may in fact be due to some type of infection. Now, one of the best tools that we have for studying immunity, again, is to look at mutants. Mutants are very important to medical biologists. And this was realized very long ago by Sir William Harvey, one of the great physicians of all time, in the 16th century, who said, "Nor is there any better way to advance the proper practice of medicine than to give our minds to the discovery of the useful law of nature, by careful investigation of cases of rare forms of disease. For it has been found in almost all things that what they contain of useful or of applicable is hardly perceived unless we are deprived of them, or they become deranged in some way." Now, I'm afraid I must have just given your translator a heart attack by putting this up. This is actually extremely difficult for even me to understand. This is old English. But what Harvey is saying here is that we don't realize how important things are, certain systems of biology are, until they go wrong, until we are deprived of them, or in some way they become deranged. So here's an example about how the derangement of the innate immune system led to a fundamental discovery. This is a paper that was published in the Journal of Immunology in 1997 about a woman who had, or a young girl who had recurrent bacterial infections. Her diseases began when she was just a little girl, when she had streptococcus pneumonia meningitis at 9 and 20 months. She had Neisseria meningitis endophthalmitis, that's an eye infection, at 13 months. She had chronic infections of her ear, chronic serous otitis media. She had an abdominal abscess when she was 3 years old that involved gram-positive diplococci. This was probably strep pneumo again. She had Staph aureus cellulitis 3 times at 3, 6, and 12 years. She has very low neutrophil counts, neutropenia, and that's persistent to the present. She had gangrene of her leg, that is a Clostridium septicum infection. She actually had an amputation because of this when she was a young girl, and she almost died of septic shock and respiratory distress syndrome at 9 years of age. She had nasal polyps. You can see that this was a very, very unhealthy entry into the world this young woman had. And surprisingly enough, no other family members in her family had any history of repeated bacterial infection. Now, we knew at the time when this was reported that this was likely to be a defect of some signaling molecule in the innate immune system. And we now know at the time, well, I should say we now know that one of the major innate immune signaling systems are these toll-like receptors. This is toll-like receptor 4, which is important in the responses to lipopolysaccharide, a molecule that comes from gram-negative bacteria. And here are all the signaling molecules that result in the production of cytokines and other pro-inflammatory genes. And you can see there are a lot that are actually on this slide. Trust me, there are about 50 times more that are not actually on this slide for you to look at. But of course I couldn't put them all here. We'd all go crazy. And it turns out that this patient, this young girl, had no IRAK4. This is an enzyme that's in the signaling cascade that begins with toll receptor activation, and it was simply the loss of this molecule that resulted in all these recurrent infections. And now we've seen several families of them with this type of disease that have been reported. Here are some of the investigators who have reported on this IRAK4 deficiency, and it gives us an insight into how IRAK4 works and the central importance of innate immunity in resolving what otherwise would be an asymptomatic infection. We also have a variety of disorders that have become increasingly studied. These are disorders that people are increasingly interested in that have been termed auto-inflammatory disorders. So these are disorders that don't require a pathogen, so they're pathogen independent. They're not due to bacteria, but they're due to mutations. There are three major forms of autoinflammatory disorders that have been defined. One's called Muckle-Wells syndrome. The second one is called familial cold autoinflammatory syndrome. And the third is called chronic infantile neurologic cutaneous and articular syndrome, or CINCA. So these go by their various acronyms and are all autosomal dominant. Now, what these disorders have in common is they have periodic fevers, rash, they have what's called amyloidosis. This is abnormal proteins that get into certain organs and cause organ dysfunction, pain in your limbs, arthritis, conjunctivitis, hearing loss, meningitis. When we say aseptic meningitis, that means that there's no pathogen involved, and cerebral atrophy. This is the type of rash that we typically see in patients who have these autoinflammatory disorders. This particular patient has Muckle-Wells syndrome. Incidentally, Dan Kastner, who gave me this photo, actually defined the word autoinflammatory disorders. And here's the type of arthritis that we might see in such a patient. Now, all of these patients have a mutation in a gene that we call NLRP3. This is part of the inflammasome family of molecules, and NLRP3 is really one of the most highly studied molecules at this time. This is a cartoon of what NLRP3 looks like, and I'm not going to go into that considering how many non-scientists there are in the audience, but each one of these lines here represents a different mutation, and really just represents a mutation in a single amino acid that is shown here. So, for example, in CINCA disorder, we see the ones in green, in Muckle-Wells syndrome, the ones in orange, and in familial cutaneous urticaria, we see the ones in blue. But the point here being that this single molecule with all these mutations results in these autoinflammatory syndromes. So NLRP3 is an immune sensor, and it controls the production of the cytokine I mentioned earlier, interleukin-1, an inflammatory cytokine. And interleukin-1 has its receptors. When it's produced, the molecule binds to the receptors, and that activates the production of certain inflammatory events, most particularly fever. NLRP3 sits in the cytoplasm of cells where it becomes activated. We're not exactly sure how it becomes activated, but when it does so, it forms a big immune complex in the cytosol cell called the inflammasome. Now what the inflammasome does is it activates a molecule called caspase. This is an enzyme, and caspase actually does nothing more than cleave preformed cytokines in the cytosol into active forms that get secreted and cause this whole syndrome. So these mutations in NLRP3 result in assembly and activation of this complex, which cleaves preformed cytokines to be released. These are cytokines that would ordinarily stay inside of cells, but instead they get out and they cause inflammation. Jörg Schaap recognized this very early. He actually coined the term inflammasome in 2001. Unfortunately, Jörg had a heart attack, a sudden heart attack a few months ago while hiking with his family in Switzerland and passed away. But Jörg had a major impact on the field. His recognition that inflammasome activation leading to IL-1 production was a very important pathological event. He concluded that patients with mutation in NLRP3 should be treated with medicines to block interleukin-1. And here's one study that he was part of where 50 patients were treated with an inhibitor of interleukin-1 known as anakinra, that's its trade name. And these are the patients early on, before treatment. This is a measure of two measures of inflammation. One is amyloid A and the other is called C-reactive protein. It's unimportant what they are. They go up whenever anybody has an inflammatory response. If you give them anakinra, these turn to normal. So this is a quote again from the famous author Lewis Thomas, who was a physician, and he says, "Our arsenals for fighting off bacteria are so powerful and involve so many different defense mechanisms that we are more endangered from them than the invaders. We live in the midst of explosive devices. We are mined." He actually thought the innate immune system had the potential to make things go quite wrong. In this case, he's actually talking about septic shock, but it refers to lots of diseases. And it's the study of inflammation that bears the dark side of immunology. Uncontrolled inflammation is linked to multiple diseases: arthritis, cardiovascular disease, neurodegenerative diseases like heart attacks, neurodegenerative diseases like multiple sclerosis, diabetes. Now it's pretty clear that diabetes involves inflammation and cancer. So again, this is not a new concept. The start of the scientific approach to understanding inflammation goes back as far as the late 18th century with John Hunter, who said inflammation is not to be considered as disease, but a salutary operation consequent either to some violence or some disease. When it cannot accomplish that salutary purpose, it does mischief. Again, this is Old English, meaning that when the inflammatory response doesn't do the right thing, it causes problems. The overall incidence of inflammatory diseases is very high in the population, and it involves major pathologic problems. Diseases such as malaria, infectious diseases, rheumatoid arthritis, osteoarthritis, asthma, multiple sclerosis, Crohn's disease, ulcerative colitis, diabetes, atherosclerosis, and psoriasis. And there are many, many types of anti-inflammatory drugs. I mean, I think you can all see how common these diseases are. If you're over the age of 30, the chances are pretty good you already suffer from one of the diseases. And if you're under the age of 30, it's coming. That's all I can say. So these anti-inflammatory drugs are really, there's still an unmet medical need for drugs that block the inflammatory response. And even, we believe, a cure for cancer may come from activating the innate immune response to fight the tumor. Consider systemic lupus erythematosus, the second most common autoimmune disorder. The first most common autoimmune disorder is rheumatoid arthritis. It's a chronic systemic inflammatory disease with an incidence of 40 to 50 cases per 100,000 individuals. One interesting side bit is that this is a very common disease in the United States. It's a very common disease here in Brazil. But in Africa and areas that have lots and lots of malaria, it turns out it's not a common disease at all. When individuals from Africa move out of the malaria zone to the United States, they have the same instance as the regular population, and a lot of people believe, for some reason, malaria protects against systemic lupus erythematosus. In any case, it's a systemic disease. It doesn't involve one organ. It involves all the organs in the body. It can cause seizures. It causes a skin rash. You can see the skin rash shown here. This is known as the butterfly rash. It causes inflammation around the heart and the lungs. And in particular, it causes chronic inflammation of the kidneys. And it's a major cause of kidney failure. Patients who have lupus, most of them will ultimately go on to have problems with their kidneys. The third most common inflammatory disease, autoimmune disease in the world is gout. I'm very aware of gout since I have it myself. And here we see the worst forms of gout. This is crippling gout. But it's been recognized for being a horrendous disorder for many, many years. This is an old painting dating from the 18th century. Really, I think that's a good description of what it feels like to have a gout attack. It is really one of the most painful forms of arthritis, and it occurs when too much uric acid builds up in the body. These uric acid crystals can deposit themselves in joints, usually in the big toe. So individuals usually present with pains in their toes. The deposits of uric acid crystals are known as tophi, and you can see them. This is tophaceous gout right here. You can see the deposits underneath the skin. And patients with gout also have kidney stones from uric acid crystals in the kidneys, and these are extraordinarily painful. It's been described since Hippocrates in the 5th century BC, and 1 to 2%, about 1 to 3% of the population has gout. So chronic inflammatory diseases like arthritis, lupus, gout are initiated when the body's own molecules are deemed to be foreign by the immune system. So in the case of gout, those uric acid crystals just don't look like they belong there, and they activate the immune response. The same receptor systems regulate this. For example, TLRs that recognize pathogens also recognize immune molecules involved in these type of diseases. So gout, amazingly enough, results from activation of the NLRP3 inflammasome that I told you a little about earlier, the inflammasome that's involved in Muckle-Wells syndrome and those other febrile diseases. And the major therapy for gout, at least for an acute attack, is colchicine. So it inhibits the ability of uric crystals to activate the inflammasome system. So what we would like when we talk about intervention in inflammatory diseases, of course, is to have drugs that hit the target on the bullseye, that are more finely tuned to inflammation and blocking inflammation. What we have is quite the opposite. We have drugs like corticosteroids, which we consider to be the sledgehammer approach to treating inflammation. Yes, those are very potent inhibitors of the innate immune response and a lot of other immune responses, but they have lots and lots of side effects, whether we're talking about the heart, the central nervous system, the gastrointestinal system, etc. The adrenal corticosteroids have terrible side effects and are very hard on our patients to take over a long period of time. What we've learned to do is to target some of our cytokines more specifically. TNF-alpha is probably our greatest success story. Neutralization of TNF-alpha can be achieved clinically with antibodies to TNF, anti-TNF antibodies. And one pharmaceutical company now markets what's known as a soluble TNF receptor. This is just a molecule that binds TNF and prevents TNF from binding to the TNF receptors on cells. So there are monoclonal antibodies, they go by these names, and soluble receptors that go by these names, and they're very, very effective for such diseases as rheumatoid arthritis and inflammatory bowel disease. Now, I already told you a little bit about anakinra and its use in autoinflammatory disorders. In addition, when we have diseases that we think are due to interleukin-1, there are now coming onto the market monoclonal antibodies to interleukin-1, as well again as a soluble receptors that can bind to interleukin-1 and trap it, known as IL-1 trap. And so when we have diseases that we think are due through inflammasome activation, these are the type of drugs that we think can help our patients live better lives. Now let me give you an example about how nicely anti-IL-1 therapy can work. Here's a small study done by Jörg Schaap, whose picture I showed you a little earlier, of 10 patients who had gout. He treated them all with anakinra or anti-IL-1, and you can see they were all symptomatic for about a day and a half, and they all got better, 10 out of 10. And I'm told that people who get anakinra think that it's truly a miracle drug. We have all of these sterile inflammatory diseases that I told you about earlier that are due to mutations in various molecules that are associated with IL-1 production, and many of these have been tested with anakinra, and to date, they've all done very well, especially the three big ones, Muckle-Wells syndrome, FCU, and chronic infantile neurologic cutaneous and articular syndrome, or CINCA. These patients, this is actually a fatal disease. These patients, this can be fatal in childhood. These patients get better immediately. I have a friend at the National Institute of Health who said he once saw a patient with Muckle-Wells syndrome get anakinra. This was a teenager who had never had really a good day in his entire life. And he said within 10 minutes or so of getting anakinra, the teenager was up, out of bed, making popcorn, and never has been the same since. He was completely cured by therapy with IL-1RA. So we think that what we do in the laboratory is worthwhile, despite what Dr. Palin has to say. Without basic research, there can be no applications. Anti-TNF and anti-IL-1 medicines were not invented by improving incrementally the efficacy of aspirin. They were invented by hard, basic scientific research that, yes, also involved studying fruit flies. We don't understand a lot about innate immunity. What we used to call nonspecific immunity has all sorts of specificities and is very complicated. When it fails, disease often follows. This is the innate immune and recognition system in somewhat more detail than what I showed you earlier. This particular slide, for some reason, didn't come out quite right, but this innate immune signaling system looks a lot like the subway map in Tokyo or New York City or Moscow. So where do we go from here? We're really not sure, but we know that inflammation is worth focusing on. There are danger-associated molecular pattern molecules that are released that cause inflammatory syndromes that are due to pro-inflammatory cytokines like TNF-IL-1, but there are other cytokines that are important as well. And the challenge is not just to neutralize cytokines, but to preserve the beneficial effects of the innate immune system, such as the host response to infection. Patients who get TNF therapy, yes, their rheumatoid arthritis gets better, but they also become at very much increased risk for infections such as tuberculosis and Listeria monocytogenes that can kill people dead. So we're very worried to use anti-inflammatory drugs of any sort without realizing that they all have complications. And in quote, in closing, I'd like to end with a quote from another American politician, possibly less known to people in this audience than Dr. Palin. This is Dr. Donald Rumsfeld. He was Secretary of Defense during the Iraq War, and he was asked at a news conference how it was that America went into the war in Iraq so unprepared for what was going to follow. And he said, "There are known knowns. These are the things we know that we know. There are known unknowns. That is to say, there are things that we know we don't know." But there are also unknown unknowns. These are things we don't know, which actually was one of the more intelligent things that, in my mind, that Donald Rumsfeld ever said. It's these unknown unknowns that are going to keep people like me and Riccardo Gazzanelli and a lot of the other scientists in the audience busy for the rest of our lives. And with that, I would also, in addition to thanking Riccardo and my other hosts here in Belo Horizonte, I'd like to thank you all for your attention.